All right, guys, so we are going to cover chapter 18 today. I'm probably going to split it into two videos like we did the previous chapter, just because I could talk about reproduction and development for hours, honestly. I think it's really, really fascinating, and it's one of my favorite chapters. So let's get started. We've got a cute little seahorse there. The fun fact, the male seahorses have a little pouch and they raise the babies, which is really cool. All right, so how do the different animals reproduce? We start off with the asexual methods. So one of the first ones up here is fission, and so you could see this cutie patootie little sea anemone. And with fission, I kind of remember mitosis when we talked about in Bio 1010 when we talked about mitosis and we talked about how the eventually the cell is just going to pinch in cleavage, 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 and then it's going to split. That's fission. That's what fission's going to do. Budding is going to be where you have your adult and the little piece just develops and grows and buds off of the adult. So here we have a picture of a hydra and its little baby budding off. And all of these asexual ones, just to remind ourselves, asexual is going to be the exact same copy of their parents. So there is no type of meiosis going on. This is just mitosis. You are making a carbon copy of mom or dad, and having it with fission and develop and pinch away, or budding and the little baby develops and buds off and goes away. Um, or it could just stick around. Sometimes they'll just stick around like sponges. Sometimes you'll just see the next one just growing and then you'll have a whole mass of it. But that's budding. We have a little video of a hydra budding in 3D. And then we have fragmentation is another type. So the story goes that the oyster harvesters, they're not big fans of starfish. So they would take the starfish and they would slice them in half and be like, haha, I killed you. But no, all they needed is a piece, a fragment. And then that fragment could reproduce. So with these guys, we have the original leg, right? And then we've got some portion of the central portion of the body, and it grew back all of the legs. So it just grew back from, we would say this guy has 5 legs, this starfish has 5 legs, and it only needed a piece of it, a fragment enough to grow a whole nother adult. And then we've got parthenogenesis, which is so cool. It's so cool. Anytime in biology, you cannot make blanket statements. You cannot say it's black and it's white. It's only, sexual and asexual, and sexual reproduction is only done in this particular way. There's no hard and fast rules in biology ever. There's always exceptions all the time. So this is asexual reproduction, and normally we'd be like, well, you have, an egg, and that egg is going to be fertilized with the sperm, and those guys, the egg is haploid and the sperm is haploid, and they come together and they fuse, and you've got a diploid zygote, and that diploid zygote mitosis, 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 and grows into me and you. But not with these guys. These guys are really cool. This is a form of asexual reproduction. So these guys grow and undergo mitosis from unfertilized eggs, So the Komodo dragon is straight out of Jurassic Park. It's really cool. And it is not fertilized. It can undergo an adult growth from an unfertilized egg. Same with bees too. Oh my gosh. So sorry for the yawning. Bees do this as well. So this is a really neat thing, and they're not the only ones. Other animals can do this too, but it's pretty rare. But parthenogenesis, I feel like, is super cool. And again, one of those blanket statement exceptions that we've got. Hermaphrodites. These are animals that are going to have both parts. They're going to have the ability to produce sperm, and they're going to have the ability to produce eggs in the same body, Not to say that they will self-fertilize. They often have the ability to still undergo sexual reproduction, and that is what they will do, but they have the ability to produce both. They can produce both eggs and sperm. All right, sex determination. We are missing the N. It's hiding behind that karyotype image. I'll have to fix that. So when we determine sex, remember I was telling you it's not hard and fast, and if you have this, you're this. Um, with peoples, with mammals, we are going to say it's down to the chromosomes, right? And so I have the image of a male and a female karyotype. So the karyotype is what happens. If you took Bio 1010, we talked about this where you take the nucleus of the cell and you bust open the nuclear membrane to get all of the genome inside. And then you're going to take that genome and you're going to stain it, and all of the different chromosomes will stain, and they'll take the stain in different colors, right? And you'll see how long the chromosomes are versus how short, where's the centromere, and then you can pair them all up, and you'll get all these homologous chromosomes that you can pair up side by side. And so if you look in this middle picture with the black background, you can see all of the human chromosomes paired together. So these are all the homologous pairs. So you've got 23 from mom and you got 23 from dad, and they are laid next to each other, each pair, homologous pair of chromosomes. And so if you look at this guy, these are all staying the exact same, and they are the same length, even though that one is a little bent and wonky looking, but these are both two X's. So we would say that this person, if they have two X's, they'd be a female. And then on this karyotype over here, we've got another X. You can check out the X. X is the default, by the way. When in doubt, it's going to be an X, unless dad contributes his Y, and then you got this. So these are the sexual chromosomes of a male. We've got one X and we've got one Y. And you can see the Y is this itty bitty little guy right here. That's the Y. So it's a little short chromosome. And on the Y is the SRY gene. And that gene is the one that, when it is expressed, is going to promote the sexual characteristics of males. But again, we can't make blanket statements. You can't make, well, you got two X's, and that's the way it's going to be, and you got an X and Y, and that's the way it's going to be. That's not true, because sometimes you could have an extra. If we talked about in Bio 1010, we talked about um, non-disjunction when we go through prophase, metaphase, anaphase, and telophase, and I've got mitosis to meiosis refreshers later on. But when we go through those guys, remember in metaphase, everybody lines up in the middle. And then in anaphase, we're going to take those homologous chromosomes and we pull them apart. And when we pull them apart, they should split equally. But sometimes that doesn't happen. And sometimes you get extra on one side and less on the other. And so you might get three um, chromosomes. You might have a trisomy going on, or you might have a monosomy and you only have one chromosome when you should have had two. So sometimes that happens, and that can happen with all of the 23 different chromosomes. You could have a trisomy or a monosomy. Sometimes that is not compatible with life, and so those embryos wouldn't make it. Sometimes the trisomies are compatible with life, and we have a trisomy 21 for Down syndrome. There could be a trisomy in your reproductive pair of chromosome 23 too. So sometimes there may be two X's and a Y. There's all sorts of crazy combinations in there. So it is not everyone's two X's and everyone's an X and a Y and that's it. It's not. It's absolutely not. You could even take the SRY gene that's on the Y chromosome and accidentally transfer it to an X chromosome. So you got an X chromosome, but you got the SRY gene on it. So there's all sorts of exceptions along the way, just to say, in general, we can generalize for sure and say two X's, female, X and Y, male. But just know that there are lots of different variations upon this. And it happens way more than you think. So that's people, that's mammals. On birds, we're going to call those chromosomes, instead of X's and Y's, we're going to call them Z's and W's. Um, some animals, their sex determination is based on temperature. So reptiles are like this. So sea turtles, we have this article from Nat Geo. I tried to find it the other day and I linked to Nat Geo, but I can't find the specific article. But it was talking about how climate change is, global warming is happening, and climate change is warming up this particular beach. And since the turtles developed their sex based on temperature, since it's so warm, all of the turtles coming from that location are all females, because that's the way turtles work. If it's warmer temperatures, it's going to be females. If it's colder temperatures, it's going to be males. I want to say alligators are the opposite. I think they are warmer temperatures are males, colder temperatures are females. So it depends on the organism. But some reptiles are absolutely temperature based, and it'll be, everybody's warm, it's this, everybody's cold, it's this. But it was an interesting article about the sea turtles, and everybody's turning into females from that beach. Um, Other organisms can change sex. Fish in particular are really good at this. If there are nothing but males in a population, they can switch to females. If there's none but females in a population, they can express genes and change sexes from female to male. So there's all sorts of crazy, wacky exceptions out there, and it's beautiful, and I love it. I love it. Okay, so we talked about determination. How do we figure out if we have the chromosomes? male or female. Then what is fertilization? So fertilization is going to be where the egg meets with the sperm. And so that could happen outside of the body, and that would be considered external fertilization. So frogs are our example here in this image. And I've got a great little video if you're curious about frog life cycle. And you could see those little fertilized eggs develop into cutie patootie little tadpoles that I currently have in my backyard in the little pond. And then the little tadpoles, the little tails will shrink up, they'll start to grow their little legs, and then boop, they're little baby frogs. So this happens outside the body, external fertilization. Internal fertilization comes in different types, which is really cool. We have the egg is going to be housed inside mom, right? And then the sperm will be donated, contributed by the male, and fertilization happens inside of the mother. But then what do we do with the development of baby? So we fertilize that egg. What happens to it? So we have different ideas here. So with this one, this is oviparity and it has ovo. So just think eggs. Think eggs. So that is when the mother has the fertilized eggs that happened inside, and then she's gonna lay the eggs. She's gonna lay the eggs and she's gonna walk away. She's gonna keep on going. No parental care. We had fertilized eggs and we laid those eggs and we are done. Our job is done. Biologically speaking, we have succeeded because in biology, all you want to do is pass on your genes and then you've succeeded. Maybe that's not your life goal, and that's okay too. So that's oviparity. It has ov in the name, so eggs. Viviparity, living. So this is us. This is us. So these are mammals. We are going to, except for echidnas and platypus, because they lay eggs. They're weird like that. But everyone else are going to have live birth, so viviparity. And then ovoviparity is an interesting combination where the babies are developing in the eggs, similar to oviparity, right? But those eggs are housed inside mom. She doesn't lay them and swim away. They're housed inside mom. So the babies are developing in the eggs that are housed still inside mom's body. So it's a combination of both ovoviparity. All right, moving on. I want to get through this section too, and then I'm gonna stop this video because the third section I feel like is gonna take a long time. All right, so fertilization, I was telling y'all, it's a process where the egg is going to meet up with the sperm. And for us, we're gonna say the egg is haploid. It only has half of the genetic contribution. So it's got half the genes from mom and it's got half the genes from dad, and then we're gonna meet up together and then we're diploid all over again, just like in that karyotype image that I just showed y'all with the 23 pairs. So we had 23 from mom in the egg, and we have 23 from dad in the sperm, and they are going to meet up, and fertilization is going to happen. So when this happens, the egg is going to come out of the ovary because the ovaries are what's going to house the gametes. We'll get into the parts later. But the ovaries house the gametes for women, and that's where the eggs are. And so on day 14, ovulation happens. That egg's going to go out. That egg is only viable to be fertilized for about roughly 24 hours. That's about the window that you have to work with. The sperm, though, live longer. Sperm can live longer. They can live up to 48, 72 hours, something like that. So if pregnancy is your goal, if fertilization is your goal, you have a very small window. You have a very small window to achieve fertilization if that is your goal. So when fertilization happens, the sperm are going to make their way up from the vagina through the neck of the uterus, which is the cervix, through the cervix into the uterus, all the way up to the top of the uterus and over the fallopian tubes to meet up with this egg. And so one of these sperm are going to be able to make it um, through all of these layers. So this outermost layer here, this stuff right here, this is the corona radiata. And so corona means crown, and it looks like it's radiating away, right? So that's the corona radiata, that's the outermost layer, and the sperm have to burrow themselves through there. They're going to have this little acrosome on the end of the head of the sperm that has enzymes that help them kind of digest their way through too. But that's the corona radiata. And then after it makes its way through the corona radiata, it has to make it through the zona pellucida. That's this inner layer here, the zona pellucida. It's got to make it through. It's got to make it through there. So it has to go through two layers, the corona radiata and the zona pellucida, and then finally it's going to make it to the actual egg membrane right there. And so once that sperm touches the membrane of the egg, and we used to just say that it was the fastest sperm, whoever was the fastest sperm made it. But now there's some research out there that shows that the egg possibly potentially chooses what sperm gets to make it through and touch the membrane of the egg. But that's fertilization. Once that sperm touches the membrane of the egg shuts down. No other sperm can come along and fertilize this egg because we are monospermy. It's one egg and one sperm. Other animals, they could be polyspermy, but we are just one egg, one sperm. Um, yeah, most sperm never make it to the egg. And then once that sperm touches the membrane, it's called a cortical reaction. Y'all don't need to know that. But you do need to know that the sperm has to make it through the corona radiata and it has to make it through the zona pellucida to make it to the membrane of the egg, which is going to lock down the egg, cortical reaction, and nothing else can come along and fertilize it. Once fertilization has happened, um, it's about 36 hours. And that one zygote. So the egg has got all the organelles that it needs in it. It's got the nucleus, it's got the mitochondria, all the stuff is inside the egg. And then the sperm's contribution is once it touches the egg's membrane, its nucleus leaves the sperm cell and goes into the egg, and then they will fuse, and then we'll have the diploid zygote. That diploid zygote is called the totipotent stem cell because it's the cell that makes all of the cells in your body, which is just mind-blowing. Um, but that's what it is. You have all the cells in your body are all built off that one cell, undergoing mitosis, 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 and different gene expression along the way. So we have this one cell, they fused about 36 hours after fertilization. It's going to undergo its first cleavage. So it's going to undergo mitosis and split into two cells. And then those two cells will split into 4 cells, and then 8 cells, and 16 cells. So this is going to be our first cleavage. So we got our 2 cells. And then when it is, oh no, that looks like it, yeah, that looks like it's 8 cells. If it's 8 cells and 16 cells, those guys are called the morula stage. The morula stage. They are a solid ball, and then it keeps going. Obviously, it keeps going. About the morula stage is if anybody has ever knows somebody participated in the process of IVF and they freeze your eggs, and then they'll fertilize them with sperm, and then they get those embryos. This is about the stage. This between about 8 16 cells. That's about the stage of the embryo development where they freeze it, where they freeze it. And then when you're ready for embryo implantation, that's about where that embryo is in development. And then hopefully, if you're doing IVF, that those little babies, those little embryos will implant and you will have a successful procedure. But that's about where that stage happens. If it's in the body, it's just going to keep trucking. It's going to keep trucking on. So 8 cells, 16 cells, 32, on and on and on. And eventually, you're going to have this trophoblast, which is a hollow ball, and the inside section is called a blastocele. Anytime you've got a section of opening, it's called a coelom. So you got the blastocele, the open space. And then this doesn't really say it, but somewhere inside here is going to be a bundle of cells called the inner cell mass. And the inner cell mass is actually what's going to be baby. That's going to be baby. So we've got cleavage is about 36 hours, and it's going to go over and over and over and over and over again, 2, 4, 8, all the way through until you've got a couple hundred cells here. And by this time, it has taken a couple days to travel through the fallopian tube and into the uterus. We need the embryo to implant in the uterus. If it does not implant in the uterus, it's called an ectopic pregnancy. An ectopic pregnancy that could happen in the fallopian tube, or it could happen in other places because sometimes the egg gets fertilized and it slips out between the fallopian tube and the ovary. That's super duper rare, but these things do happen. So we want the embryo to implant in the uterine wall. That's where it needs to be. As it's journeying through the fallopian tube and going down, it's going to implant at about this stage here, and then it's going to undergo this process called gastrulation. So that inner cell mass I was telling you about, that's going to start to fold in on itself, and then we are going to end up with a gastrula. And at the end of this stage, we are starting to form the germ layers. So if you want to go from the outside in, is what I find the easiest. Ectoderms on the outside, Mesoderm's on the middle, and then endoderm is on the inside. So those are the three germ layers. Here's a picture that I steal from my anatomy and physiology class on reproduction, and you can see this embryo has implanted. This is going to be the uterine lining, which is where we want the embryo to implant. When that embryo implants, you remember I was telling you that there's a ball and there's an inner cell mass. So that hollow ball secretes a whole bunch of enzymes. This is the hollow ball on the outside, secreting a whole bunch of enzymes to help it implant into the uterus. So it's going to like burrow its way through the uterine tissue and burrow in there. And while it does this, it's also secreting chemicals to trick mom's body because otherwise mom would be like, what is this parasite? I need to get rid of it. And the embryo is releasing, I can't remember what the name of it is. It was something with numbers and letters. But it releases a chemical to trick mom's body. So it doesn't realize that this, honestly, it's a parasite. It doesn't realize this parasite is there in mom's body. So we have the beginnings of the germ layers starting to form here and here. And then eventually we're going to get all three germ layers. So here's our ectoderm still in blue. Here's our endoderm. And then this stuff will be our mesoderm, everything in the middle. This little yellowy sac here, this guy, I don't have time doodling. That guy is going to be our yolk sac, and then this guy is the amniotic sac. So there's two different sacs during pregnancy. There's the embryo, the yolk sac that's going to feed into the embryo and be part of your GI tract, and it's going to help feed. Like if you enjoy scrambled eggs, the yellow part is the yolk, right? We have the same thing, same thing. But then we also have the bag around baby. We can't forget about the bag around baby, and that's the amnion. So that's this little blue section here. So anytime you watch TV and somebody's water burst, it's the amniotic sac, it's the bag around baby, which is a really cool feature that we talked about previously when we talked about how reptiles were able to move away from water. It's because of this, it's because of that. Here's another close-up of it. And then here's our three germ layers, ecto, endo, meso in the middle. This is that yolk sac getting absorbed in and becoming part of the GI tract for us. These are the three germ layers, endoderm. I'm just going to say GI tract, digestive system. I'm not going to ask y'all about any of the other things. Mesoderm is in the middle, muscles, bones, circulatory system is mesoderm, everything in the middle. Ectoderm is everything on the outside I was talking about, so hair, skin, nails. It also includes your nervous system, which is really cool because when we talked about that inner cell mass, the little flat bit of cells that's inside the ball, it's flat. It's flat, and that's going to be the ectoderm, and that folds in on itself and forms the neural tube as well. So your central nervous system and your peripheral nervous system is all coming from that same tube structure in the very, very beginning, and that's your ectoderm. That's your ectoderm, which is cool. More germ layer stuff. All right, so I'm going to finish this video here and then start the 8.3 section, which is a longer section in the next video.